Hi guys, so we're here today for DNA replication. And you can see we're starting with our chromosome and how we take and unwind our chromosome, ultimately unwinding and opening up the DNA to make the new strands. Just pictorially, that's kind of showing you some of these things we've talked about. And we have, notice our adenine and thymine pair together and our cytosine and guanine pair together. Okay. So when we talk about DNA replication, you have to think back to when we learned about mitosis and interphase specifically in the cell cycle. During S phase of interphase of the cell cycle is when DNA actually replicates, and this has to happen before we can go through the mitosis process to divide the nuclei, which of course happens before we can go through cytokinesis. So the question is, how exactly does it replicate? What happens? And there were a lot of scientists that were trying to figure this out for a long time. So they came up with three kind of key alternative models for DNA replication. A conservative model, where we conserve or keep the original and then end up with the original and a brand new copy. A semi-conservative model, you'll see here we have part of our original goes to each of the new ones, and then we have a new other half. And then we have the dispersive. So you'll notice it's kind of a mixture of new and old, just kind of randomly mixed together. Okay, so let's look a little more detail at this conservative model, where we have our parental strand, the one we start with, and we end up with the parental strand and a new strand that's supposed to match it. So the idea is that the parental strand will direct the synthesis of an entirely new model of double-stranded molecule. So the parental is the pattern, but we make a whole new one. So the parental strands are fully conserved. That's where the name conservative model comes from. Okay, then we have our semi-conservative model. Okay. So the two parental strands each will make a copy of themselves. So we have an original parental and we have a new strand in each of the processes. So after one round of replication, the two daughter molecules will each have one parental, that's our gray, and one new, that's our blue. So we've sort of conserved it because we kept half of the original and then the other half's new, hence semi-conservative. And then our third one is the dispersive model, and this is the one where we kind of have a little mix and match. So the material of the two parental strands is dispersed randomly between the two daughter molecules. So we end up with just layers upon layers upon layers, where some is from parental and some is from the new. So after one round of replication, the daughter molecules have a random mix of the parental and the new DNA. So those are the three models that basically were brought up and being studied. So the question is, which one's correct? Because I guess we could argue any of them. So in 1954, Messelson and Stahl performed an experiment using bacteria. Specifically, they used E. coli, which, remember, we always hear about E. coli as a negative, but we have E. coli in our systems, so not all E. coli are going to kill us or make us sick. Bacteria was cultured with a heavy isotope known as nitrogen-15. So we have E. coli with nitrogen-14 and the special E. coli with nitrogen-15. So we transferred into the medium that has nitrogen-14. Okay. So then DNA was then centrifuged and analyzed after each replication. And so when they started at 0, we had only nitrogen-15. And then as we progress, you'll notice that we keep getting less and less, but we always have nitrogen 15 and we have nitrogen 14. So every time we're replicating, we're pulling in that nitrogen 14 that wasn't there originally, that we've stored it in. Okay, so they took samples of DNA after each generation, and they found that the parental strands were following the semi-conservative model. That's why we always still have some of the nitrogen 15 strands, but we keep getting more and more and more of the nitrogen 14 as we're making new each time. So this showed us that the semi-conservative model was the model that DNA replication seems to follow. Just looking at it pictorially a different way, so you can see we started with 2 of the N15s. They each made a copy where we had 1 N15 and 1 new, which was 14. And then each of those made a copy. They split and copied. And so you see how we keep getting more and more of the N14, but the N15, that original parental strand, still keeps showing up. That's why we keep seeing it, but it's a smaller percentage each time. So that's how they determined that it was the semi-conservative model. So then looking at DNA replication, there are specific steps and parts involved. So first step, DNA replication begins at sites that are called origins of replication. So there's a specific point at which it will start. Different proteins are going to attach to the origin of replication. They're going to open up the DNA to form what we call a replication fork. Now notice this fork only has 2 prongs because we have 2 strands. Then an enzyme called helicase, I know it's an enzyme, you can see the ASE, will unwind the DNA strands at each replication fork. So this is where that hydrogen bonding comes in handy, because it makes it easy for helicase to unwind the DNA strands at that replication fork. To keep that DNA from rebonding with itself, we use specific proteins called single-strand binding proteins, SSBPs. They're actually going to bind to the DNA so they keep it open. The idea is we're sticking a piece in there so that it can't join back together because there's something blocking it. So topoisomerase, sorry, sometimes I got to work on my tongue twisters, will help prevent strain ahead of the replication fork by relaxing the supercoiling. So if you think about it, if we have something all coiled up and you start pulling it apart from one end, it kind of tightens up the coil behind it normally. So we use this special enzyme to relax that coil so it's not getting tighter and tighter and tighter so it doesn't break. Because the idea is if it got strained too much, it might cause it to break. So here we put the helicase in. Helicase is opening it up, breaking those bonds. We're going to add the SSBPs, and these are, remember, they're blocking it, kind of holding it open so it can't close. And then we're relaxing this coil because as we're pushing it open, we would be pushing and tightening that coil. So that's what our enzyme's coming in here to relax that coil so it doesn't get too. 3, the enzyme primase, prime as in beginning, primary, primase initiates replication by adding short segments of RNA, which we call primers, to the parental DNA strand. So there's these little sections that are going to match up with those initial DNAs. So if the initial DNA was TTC, then this little RNA primer would have AAG to match up with it. So it's kind of like putting a little start piece in there. The enzymes that synthesize DNA can only attach new DNA nucleotides to an existing strand of nucleotides. So that's why we have to put those primers in, because we've got to have a base to start building off of. If we don't have the foundation, if we don't have the primers, we can't build the rest of the walls, the DNA strand. So the primers are the foundation. So here we go. This is what we've been working on. So we're going to add our little RNA primer. That's our little start molecule. That's our foundation that we're going to build on. And then we have what we call anti-parallel elongation. So DNA polymerase 3 specifically attaches to each primer. So we put the primer on and we're going to add this little enzyme called DNA polymerase 3. It's going to attach to the primer on the parental strand and it's going to move in the 3' to 5' direction. But remember, we had anti-parallel strands, so they were opposite each other. So 3' to 5' on one side moves in one direction, 3' to 5' on the other side moves in the opposite direction. So anti-parallel elongation. So as it moves, it adds nucleotides to the new strand in the 5' to 3' direction. Because if we started at 3' on the parent, that would be the 5' on the new daughter strand. It will follow the helicase, and it is known as the leading strand and only requires one primer. On the parental strand that moves the opposite direction, the lagging strand, we're going to have many primers because we have to build it in little pieces as it's moving away from the helicase in little segments. Another polymerase on this one, and we'll build that way until we can come up here and then we will be able to join these little pieces. And so as this continues to move to the right, unwinding, we'll add another primer and we'll keep working the lagging strand in pieces. But the leading just keeps going. Okay, so the leading strand is synthesized in one continuous segment. But the lagging strand moves away from the replication fork, so it's going to be in little chunks. We call these chunks Okazaki fragments. Those are those little segments of the lagging strand. Yes, you do need to know that. Okay, so we're building. See how that went continuous on the leading strand, but on the lagging strand, we have to do it in small sections. And that's the Okazaki fragments. After we form those Okazaki fragments, we have DNAP1, so it's another primer, replaces the RNA nucleotides with DNA nucleotides, then being able to connect where that RNA primer was. We replace those little pieces with DNA and we can connect all of these little fragments. So we'll use DNA ligase. When we ligate something, we're joining them together to form a continuous strand. So we've got our fragments. DNAP1 replaces the primer. Does the same thing on the lagging strand. It's replacing the RNA with DNA. And then we have the ligase that's going to connect each of the fragments together. Okay. So there are YouTube videos and that I will make sure videos. So sometimes we run into problems at the 5' end. Since DNAP3 can only add nucleotides to a 3' end, there's no way to actually finish the replication on the 5' of that lagging strand, because remember, that's the one we're doing in little strips or pieces. So over many replications, the DNA is going to get shorter and shorter and shorter. That would be a problem. So how do we protect the genes on that DNA? So at these ends, we have what we call telomeres, repeating units of short nucleotide sequences. They don't code for anything. They're just there. And so every time we do this, we may lose a little bit, but we have all this extra, so we're not actually losing any critical information. That's the point of the telomeres. So it's kind of like putting a little cap end on the DNA to put off that erosion. But ultimately, when these telomeres start affecting genes, that's where we really start to see the aging process. Telomerase can add telomeres to DNA, so over time, we can replace the ones that are being damaged or discarded to a certain extent. Okay. Additionally, if you've ever tried to just copy a sequence of letters, sometimes you make little mistakes. Well, the same thing happens here, so we always want to proofread and repair any damage that may occur. So as DNA polymerase adds nucleotides to the new strand, it's also proofreading the bases it adds. So it's double checking and says, hey, wait a minute, is this right? Is this not right? If there are errors that occur, we have what's called a mismatch repair that's going to take place. So enzymes will actually come in, remove and replace the ones that aren't paired correctly with a correct pair. If the segment of DNA is actually damaged during this process, nucleases can remove the entire segment, and DNA polymerase and ligase can come back and replace that segment that was damaged. So if it's just a little bit, we can often repair it. If it's a bigger section, we may have to cut out, remove the chunk that's damaged, and then replace it. So quick review. If a parental DNA strand reads ACGTAC, what would the newly synthesized complementary strand read? You always want to think what pairs with what pairs with. So A pairs with T, C pairs with G, right? So TGCATG would be the correct strand, and notice that the directionality is opposite. So if this one was 5' at the beginning, this one's 3' at the beginning. Summarize the role of helicase, topoisomerase, and DNA polymerase 3. Okay, so helicase unzips the DNA, topoisomerase helps reduce strain, DNAP3 adds nucleotides to the new strand of DNA. What direction does DNAP3 move along the parental strand? It always moves 3' to 5'. Why do Okazaki fragments occur on the lagging strand? Remember, that's the one where the fragments occur because it's moving away from the replication fork, so it has to replicate in small chunks. Then we have to go back